Welcome everybody, I'm Matteo Brunetti and I'm here to present you this study on the cryptic diversity in a phytophagous insect species, Psylliodes chalcomera. Psylliodes chalcomera is a Palearctic beetle species belonging to the family Chrysomelidae, a family that includes only exclusively phytophagous species. Adult individuals of Psylliodes chalcomera shows a wide variability in dorsal coloration, and this fact led to the description of some chromatic forms, but none of them have been attributed taxonomic values. These species usually occur in open environments, feeding on several species of thistle, so Asteraceae, and it is a univoltine species. Each female lays about 300 eggs near the host plant during fall and spring. The hatched larvae are minors, so they feed inside the rosette and stems of the host plant until pupation. As you can see in this picture, where the larva is captured inside the mine in the stem of the plant. Adults usually emerge in early summer, and differently from larvae, they feed on host plant leaves. During the hottest period of summer, when most of the host plants are dried, those individuals enter in a diapause state until November, when mating occurs. As I said, Psylliodes chalcomera individuals feed on different plant species belonging to Asteraceae, but the main host plants are those three reported here. So species belonging to the genus Carduus, mainly Carduus nutans, and Centaurea solstitialis, and species belonging to the genus Onopordum, almost exclusively Onopordum acanthium. Previous studies tested the specificity of the association of Psylliodes chalcomera with the host plant, and they performed choice and no-choice experiments with individuals collected on different host plants, on those three specifically. The results show a strong specificity for the host plant in almost all the cases, while only individuals collected on Onopordum were able to feed also on Centaurea solstitialis, but their embryos did not survive. Given the strict association with the host plant, the internal and external morphology of the insect have been studied, looking for characters that allow to distinguish insects associated to different host plants. But the morphological variability of those insects associated with different host plants completely overlap, and no character were unique to a single group. In order to further characterize the three suspected biotypes of Psylliodes chalcomera associated with different host plants, we planned this study that aims to investigate the genetic diversity of population of Psylliodes chalcomera associated with different host plants and collected in several areas of the native range of this species. So 159 individuals, 67 larvae and 92 adults were collected from several localities within the native range of Psylliodes chalcomera. Adults were collected individually on plants that had adult feeding damage using an entomological aspirator, while larvae were collected by dissecting the stems of infested plants. So all the collected individuals were morphologically identified as belonging to the species Psylliodes chalcomera. Then the total genomic DNA have been isolated and a fragment of the CO1 mitochondrial gene has been amplified. Several tools have been used to characterize the genetic diversity of the sample population, including a phylogenetic reconstruction obtained through Bayesian inference. And this is the maximum clade credibility tree obtained from the Bayesian phylogenetic inference. Each tip corresponds to a CO1 haplotype, and the number of the nodes on the nodes represents the posterior probability of the clade. As reported in the legend, the first column of colors correspond to the host plant, while the second to the country in which the insects were collected. The two main clades that we can identify are quite well supported, but do not correspond to a pattern determined by the host plant. So the first group includes individuals collected both on Onopordum and Centaurea solstitialis. This plant is commonly called the yellow star thistle, so we call this group OYS, Onopordum yellow star thistle. The second group that is composed by individuals collected mainly on Onopordum and Carduus, so it has been called OCA, Onopordum Carduus, but also a few individuals collected on Centaurea solstitialis. Within these main clades, other groups with quite high support can be identified. The first is composed by almost exclusively by individuals collected on Centaurea solstitialis, but it also includes an haplotype associated to Onopordum. The second group contains individuals associated to both Centaurea solstitialis and Onopordum, mainly Onopordum, while the third contains only a haplotype associated to Onopordum. The fourth group corresponds to the OCA group, since its subgroups are not well supported. As you can see, those groups seem to better mimic the ecological pattern determined by the host plant, but often there are few haplotypes associated to a plant that is different from the one typical of the group. We hypothesized that this noise could be due to the inclusion of both larvae and adults in the dataset, and this for two main reasons. Firstly, it is a well-known phenomenon that adult phytophagous insects can have a wider range of host plants when compared to the larvae, even when the strict association of the larvae with the specific host plants can act as a post-zygotic reproductive barrier. Moreover, the association with the plant is certain for the minor larvae that are collected inside plant tissue, but it could be wrongly seen to adult insects that are highly motile and able to fly, so when other plants are present in the surrounding of the plants on which they are feeding, they could be collected on a different plant just due to chance, not because they are really eating that plant. So we reanalyzed the dataset, keeping only sequences belonging to larvae, and this is the tree that you obtain. Colors and legends are the same of the previous slide. As you can see, now the ecological pattern determined by the host plant is definitely clearer, and the support on the nodes are higher. The group and the identified group are almost the same, but group 4 is missing. Group 2 is missing, since we do not have larvae individuals for the population in this group. Now, group 1 and 3 clearly correspond to individuals collected to a single host plant taxa, with group 1 including individuals collected only on Centaurea solstitialis, and group 3 individuals collected only on Onopordum. Instead, group 4 is composed by haplotypes associated to both Onopordum and Carduus, and there is even a single haplotype that is associated to both these host plants, the last one at the bottom of the tree. Almost the same grouping pattern can be visualized in the haplotype median joining network. On the left side, the colors in the network correspond to the host plant, while on the right side, colors correspond to the collection country. Comparing the two figures, it is clear that the main group are more strictly related to the trophic ecology of the insect than they are with the geographic distribution. As an example, we can see that haplotypes colored in red in the right side network, so those collected in Russia, are splitted in two groups that correspond to the group 1 and 3 that are determined by the ecology. Genetic distances computed on the CO1 region are usually used to determine the limit between intraspecific and interspecific clades. In our case, most of the distances within the group have a range comparable to between group distances, ranging from 0.2 to 0.5%. Only group 4 exhibits higher distances, of about 0.8, 1.8% from other group, but those distances coincide with the optimal barcoding threshold inferred for Alticini tribe of Chrysomelidae, and so it is difficult to define them as inter or intraspecific, but to be conservative, it is probably better to consider them as intraspecific. So, resuming what we know, Psylliodes chalcomera is composed by at least 3 genetically distinguishable biotypes that have highly overlapping distributions and are characterized by different host-plant preferences. Group 1 is associated to Centaurea solstitialis. Group 3, and probably also Group 2, even if we don't have a larva specimen for this group, are associated both to Onopordum, while Group 4 includes haplotypes associated to both Onopordum and Carduus. These 3 biotypes are morphologically identical and no internal or external character permit to distinguish them. Their distribution is highly overlapping, and in some localities 2 biotypes occur in complete symmetry. Even if Group 3 and 4 feed both on Onopordum and Group 4 feed on 2 different plant genera, the specificity for the host plant has been proven to be really high in experimental tests. The identified group are well supported by phylogenetic analysis, but the genetic distance among them should be still considered as intraspecific. So, the identified biotypes probably are an example of within-species divergence that could eventually lead to a future speciation event. The ecological specialization, in this case the specialization towards a host plant, is already present, so predating the hypothetical complete divergence that would lead to a speciation event. So Psylliodes chalcomera could represent strong evidence of the contribution of host shift to speciation in phytophagous insects. It is an highly debated topic within the topic of ecological speciation. Nevertheless, further studies are needed to better define the degree of genetic differentiation among these lineages of Psylliodes chalcomera. Thank you all for your kind attention, and I would like to thank also all the other authors of this study that are listed here, and I'm here to answer any question you have.